Welcome to the deep dive. Today we are tunneling past, the magic of sunlight and photosynthesis, and we're plunging right into the secret life happening under the soil. We're exploring what plants actually need to thrive, the real physics and chemistry of dirt, and maybe more importantly, why plant growth is almost always limited by this hidden hunger for specific minerals.
That's such a crucial point. I mean, when you look at a huge oak tree, something like 96% of its dry weight is just carbon, hydrogen, and oxygen, stuff it gets pretty easily from air and water.
Right.
Our deep dive today is all about the other 4%. And that tiny fraction, that's where all the action is. It's made of inorganic minerals pulled from the soil, and it literally dictates whether that plant lives or dies.
So our mission today is to give you a complete roadmap of plant nutrition. We're going to start on the ground floor, literally, with the structure and chemistry of the soil. Then we'll pinpoint the 17 essential elements plants can't live without. And finally, we'll get into the incredible partnerships plants form with microbes to get those resources.
It's amazing, isn't it? That 4% strategy involves fighting gravity, managing electrical charges, and even hiring bacteria to do impossible chemistry.
Okay, so let's unpack the soil itself. We tend to think of it as just dirt, but it's this highly layered environment. These layers are called soil horizons.
That's right. And while the deepest sea horizon is mostly just broken down rock, the real action, as you said, happens in the A horizon. That's what we all call topsoil.
And topsoil is everything to a plant.
It's its entire lifeline. It's where rock has been broken down and mixed with air, water, living things, and importantly, decaying organic matter. I mean, to give you a sense of scale, a single teaspoon of healthy topsoil can have billions of bacteria, plus fungi, protozoa, all sorts of things.
So the physical makeup of that topsoil, its texture, that's defined by particle size, right?
Well,
Sand being the biggest, then silt, then clay.
exactly. And we hear about perfect dirt, but what that really means is an ideal soil we call loam. Loams have a pretty equal mix of sand, silt, and clay, and that balance is key. You need the sand for aeration so the roots can breathe and water can drain, but you need the silt and clay to hold on to that water, and crucially, the dissolved nutrients.
If it's all sand, everything just washes away.
Right. And if it's all clay, the roots literally drown. There's no air.
And what about the organic stuff? As things die and decay, they form humus. That's not just filler, is it?
Not at all. Humus is chemically active. It's vital. It helps the soil hold water. But its most important job is probably its role in coing exchange.
Coing exchange. Okay, let's break that down.
So this is the big chemical challenge. Most of the particles in good soil, especially the clay and the humus, have a net negative electrical charge.
Yeah, I think I see the problem. If the soil particles are negative, they can't hold on to other negative things.
Precisely. So the negatively charged nutrients, the anions like nitrate or phosphate, they get repelled. They just stay dissolved in the soil water, which means they get washed away or leached really easily when it rains.
So the plant loses out. But the positive ions, the cations, potassium, calcium, magnesium, they stick like magnets to the negative soil particles.
They do. So the question becomes, how does the plant get something that's basically glued to the dirt?
And this is where that cation exchange comes in.
This is it. It's an incredible chemical trick. The plant can't absorb the cation directly from the soil particle. It has to be floating free in the soil solution in the water. To make that happen, the roots actively make the soil right around them more acidic.
Wait, really? Plants are creating their own little acid bubble just to eat. How do they manage that?
Mostly through respiration. The roots release carbon dioxide, CO2. That reacts with water in the soil to form carbonic acid, which then releases hydrogen ions, H+. They also just pump H+ ions out directly.
So these hydrogen ions flood the area.
And because there are so many of them, they just physically knock the mineral cations like potassium off the soil particles.
And once the potassium is floating free in the water, the root can finally absorb it. That's so much more active than I imagined.
It's a fantastic mechanism, but it completely depends on that topsoil being stable. And that brings us to how we, as humans, have mismanaged it. The American Dust Bowl in the 1930s is the textbook example. Deep plowing exposed the topsoil, and it just blew away.
And that kind of mismanagement is still happening, especially with water. The sources say 75% of all global fresh water goes to agriculture. A lot of that from aquifers.
And those aquifers are deep, ancient sources. When we drain them too fast, we get two huge problems. First, they just run out. But second, you get land subsidence. The ground itself actually sinks when the water is removed. It's a massive issue in places like Florida and California.
Irrigation also has that hidden danger of soil salinization, right? Where the salts in the water are left behind and build up in the soil until it's toxic.
Exactly. And then there's fertilization. You see the NPK ratio on a bag of fertilizer, say 15-10-5. That's nitrogen, phosphorus, potassium. The problem is these commercial fertilizers are immediately available. If the plant doesn't grab them right away, those anions, especially the nitrate, get leached into waterways and cause these huge destructive algal blooms.
Which is why organic fertilizers like compost are often better, because they release the nutrients slowly over time.
It mitigates a lot of that immediate runoff risk, yes.
Before we move on, I wanted to touch on this really cool biotech solution called phytoremediation. Instead of using harsh chemicals to clean up polluted soil, you can just use plants.
Like a biological vacuum cleaner.
Exactly. The example given is the alpine pennycress. It naturally pulls up and accumulates toxic metals like zinc in its shoots. So you plant it, let it grow, harvest it, and you've effectively removed the zinc from the soil.
It's a brilliant non-destructive approach, and it brings us right back to that critical 4% of the plant's mass. Let's define what we mean by essential. An element is only essential if a plant absolutely needs it to complete its life cycle, to grow and make seeds.
Okay, so how do scientists figure that out? You can't just look at the soil because it's full of everything.
You can't, so they use a technique called hydroponic culture. They grow plants with their roots suspended in water in these aerated mineral solutions. Then they can create a solution that has everything except one element. If the plant fails to grow or reproduce, they know that one missing thing was essential.
And through that process, they've found 17 essential elements for all plants.
17, yes. And we group them by how much the plant needs. First, you have the macronutrients, 9 of them, required in pretty large amounts.
So if we take out carbon, oxygen, and hydrogen from air and water, that leaves 6 major ones from the soil.
Right. The big 6 from the soil are nitrogen, phosphorus, sulfur, potassium, calcium, and magnesium. Nitrogen is, hands down, the single most limiting nutrient for plants worldwide. It's a core part of proteins, DNA, chlorophyll, everything.
And then phosphorus is the energy currency, ATP, DNA, cell membranes. Potassium is what controls the stomata, the pores in the leaves.
It's a major player in yes, it regulates the turgor pressure that opens and closes them.
Then you have the 8 micronutrients, things like iron, manganese, zinc. Why does the plant need such tiny, tiny amounts of these?
Because their main job is catalytic. They function almost entirely as enzyme cofactors. They aren't part of the plant's structure. They're like the keys that turn on the chemical machinery. A tiny amount of a key can start a huge engine, you know?
That makes perfect sense. So if a plant starts to look sick, say its leaves are yellowing, a condition called chlorosis, how do you diagnose what's missing?
You have to look at where the symptoms are showing up first, and that all depends on whether the nutrient is mobile inside the plant. Yeah, can the plant move it around? If a nutrient like nitrogen or magnesium is mobile, the plant is smart. It'll actually cannibalize its older leaves and move that limited supply to the new, young, growing shoots, because that's where reproduction happens.
So if the older leaves are turning yellow first, it's a mobile nutrient deficiency.
Bingo. And conversely, if a mineral is immobile, like iron, the plant can't move what's already there. So the deficiency symptoms show up first in the younger leaves, while the old leaves stay green. It's a really critical diagnostic clue.
So for anyone listening who gardens or farms, if you see maize, for instance, with yellowing starting at the tip of the oldest leaves and moving down the middle, that's a classic sign of nitrogen deficiency.
Exactly. Or reddish-purple edges on the young leaves. That's screaming phosphorus deficiency.
So we've covered the physics and the chemistry. Now let's get into the biology.
Yeah.
The alliances plants make.
Right, because plants dedicate a huge amount of energy to this. It's not a passive process.
It really isn't. The source material says a plant might secrete up to 20% all the sugar it makes through photosynthesis, just to feed the bacteria living on and in its roots, the rhizobacteria and the endophytes. So what does the plant get back for that massive 20% investment?
It gets protection from pathogens, growth hormones, and most importantly, access to nutrients it could never get on its own. And the biggest one is nitrogen.
The nitrogen cycle.
The big one. The air is almost 80% nitrogen gas, N2, but that triple bond holding it together is one of the strongest in nature. Plants can't break it.
So if you can't do the job yourself, you hire a specialist.
You hire a bacterium. The process is called nitrogen fixation, converting that unusable N2 gas into ammonia, NH3, which then becomes ammonium, NH4 plus N-Sali in the soil. Only certain bacteria can do this using a special enzyme called nitrogenase. And it is incredibly energy intensive. Costs 16 ATP molecules for every tiny bit of conversion.
And once the ammonium is there, other bacteria step in to convert it to the form plants like best.
That's right, a process called nitrification. They oxidize ammonium first into nitrate and then into nitrate, NO3. That's the form most plants can absorb most easily. And of course, you have a modification where decomposers break down dead things and return their nitrogen to the soil as ammonium. It keeps the whole system going.
And the most famous example of this partnership has to be between legumes, beans, peas, peanuts, and rhizobium bacteria. The plant literally builds them a house.
It does. It builds a nodule, a swelling on the root where the bacteria can live safely inside the plant's own cells as bacteroids. But this creates a huge chemical paradox.
Which is?
The enzyme nitrogenase that does all the work, it's instantly and permanently destroyed by oxygen. But the bacteria need oxygen for respiration to make the massive amounts of ATP needed to power the reaction.
That's a biological catch-22. You need oxygen to make the energy, but the oxygen destroys the machine that uses the energy.
Yeah.
Exactly. And the solution is mind-boggling. The nodule produces a protein called hemoglobin. It's a reddish molecule, and it's chemically almost identical to the hemoglobin in our blood. It acts as an oxygen buffer. It grabs onto oxygen, keeping the concentration just high enough for the bacteria to breathe, but low enough that the nitrogenase enzyme is safe.
A plant is making something that's basically plant blood just to keep its bacterial partners happy. That's an incredible level of cooperation, and that's the whole principle behind crop rotation. You plant legumes to naturally put nitrogen back into the soil.
That's the sustainable way to do it. And moving from bacteria to fungi, we have another huge partnership called mycorrhizae, literally fungus roots. Over 85% of all plant species do this.
What's the deal here? What does the fungus bring to the table?
The fungus gets sugar from the plant. In return, its web of filaments, the hyphae, acts as a massive extension of the plant's root system. It dramatically increases the surface area for absorbing water and minerals, especially phosphorus, which can be really hard for plants to get.
And there are two main types of these mycorrhizae with different structures.
Yes, the first is ectomycorrhizae. You see this mostly with woody plants like pines and oaks. The fungus forms a thick sheath, a mantle, over the outside of the root. The hyphae then grow into the root, but they stay between the cells in the extracellular space. They never go inside.
And the second, more common type.
That's arbuscular mycorrhizae. Here, there's no sheath on the outside. Instead, the fungal hyphae grow into the root cells themselves. They penetrate the cell wall, but not the plasma membrane. They just kind of indent it, like pushing your finger into a balloon. And inside, they form these tiny, intricate branching structures called arbuscules, little trees, and that's the main site where nutrients are exchanged. It maximizes the surface area for trade.
So just to wrap up, we should probably mention the real outliers, the plants that have evolved these bizarre strategies for nutrient-poor environments.
Absolutely. You have epiphytes like staghorn ferns. They just grow on other plants, but they're not parasitic. They get their nutrients from rain and dust.
Then you have the actual parasitic plants, like dodder, that use these special roots called haustoria to just tap directly into the host plant and steal its nutrients.
And of course, the most dramatic ones, the carnivorous plants. Venus flytraps, pitcher plants, they still photosynthesize, but catching and digesting insects is a way to supplement their diet, especially with nitrogen, in soils where there's almost none available.
So we've really covered a huge amount of ground today. We went from the ideal texture of loamy soil and the chemistry of cation exchange all the way through the 17 essential elements and down into these incredibly complex mutualisms like nitrogen fixation and mycorrhizal networks.
It's a journey from the macro scale of soil structure right down to the molecular level of hemoglobin buffering oxygen. It's just fantastically complex.
It really makes you rethink what a plant even is. We define them by their ability to feed themselves through photosynthesis autotrophy. But the sources hint that some common plants, like Arabidopsis and even some tomatoes, might be capable of a limited kind of carnivory, trapping and digesting bacteria and yeast.
And that raises a fascinating question for the future of agriculture, doesn't it? If even our standard plants have this hidden flexibility, this ability for limited heterotrophy, how much do we still underestimate them? What other surprising nutrient sources might we be able to unlock, maybe by engineering these microbial partnerships? It could be key for sustainable farming in the future.
A really compelling thought to leave you with. Thank you so much for joining us for this deep dive into the 4% that rules the entire plant kingdom. We hope you feel thoroughly informed and maybe you'll look at the soil beneath your feet with a whole new appreciation. From all of us at Last Minute Lecture, thanks for listening.